Hi everybody, and welcome to your first AP Bio lecture. We're going to continue moving on with our chapter, and we're going to talk about cellular organization, so how cells are organized in a meaningful way that allows for their survival. So what if I told you that you don't contain cells, but you're actually made of cells? So this is a big misconception that people have. You hear a lot of people say that you contain trillions of cells, when in reality, you are actually made up of trillions of cells. And this is a pretty big distinction. So it's like talking about a box of Legos. So here we have a box that contains 650 pieces of Legos versus a Lego box, something that's constructed or made entirely of Legos. And so we are actually more like this Lego box. We are made of cells. When we talk about the basic cellular organization, we're going to focus on the membrane, the nucleus, the cytoplasm, and the organelles. So because we are made entirely of cells, we have lots of different types of cells that have very specialized functions. In order for those cells to work the way they're supposed to, they have different parts that give them different characteristics depending on what kind of cell you're looking at. Here we have just a very classic picture of an animal cell and all of the different features. And here's a plant cell. So just by comparing these two cells, you can tell that they probably serve very different functions because of the different structures they have. Both types of cells will have a cell membrane, and this is going to be what separates the cell from the environment. So if you're comparing a cell to, let's say, a person, it would sort of be like our skin. This is the boundary layer around the cell, and it's important for controlling essentially what moves in and out of a cell. Just inside that cell membrane, you have the cytoplasm or the cytosol, and this is going to be the substance essentially between the membrane and the nucleus. It's what we like to call the fluid part of the cell. What I do want you to keep in mind, I know that a lot of people will hear things like the cytoplasm is the jelly within the cell. And what we're going to talk about later on when we get to the cytoskeleton is that the inside of a cell isn't necessarily like jelly. There are different structures within the cell that help it to move, that help it to maintain its actual form. And so don't try not to think of it in that term necessarily, but it is sort of the more fluid part. It is within the cell membrane. That's where you're going to find your organelles. They're going to be located within the cytoplasm of the cell. And so when I talk about an organelle, it very much is like the organ in our body, so a small organ. And it's going to be something that has a very specialized function that's important for cellular organization. So all the organelles that we talk about all serve a different purpose within the cell. These are essentially compartments that separate the different chemical reactions. So each of these organelles is responsible for something in the cell, and so you want to keep those reactions separate. Everything, if you look at our bodies, our heart is, separate from, let's say, our stomach. Those two perform very different functions, so they're kept separate from each other. With this chapter, you guys are going to be responsible for being able to identify the major organelles, and you need to be able to give their structure and their function. So kind of keep that in mind as we go through. The first thing we're going to talk about is the nucleus. This is the most conspicuous organelle. It's going to be the one that you can see with the microscopes that we have here at school. It's usually pretty spherical. It can, however, be irregularly shaped. And with the nucleus, you are going to be responsible for a few other structures. So there's a nuclear membrane, again, a layer that separates the nucleus from the rest of the cell. That membrane has pores. Just like your skin has pores, this membrane is going to have pores as well to allow movement of materials in and out of the nucleus. There's also a nucleolus as well as chromatin. And so what you're looking at here is just a diagram of a nucleus. You can see we have the outside, or the nuclear membrane. You've got some nuclear pores that you can see, so those are marked by the red. Inside, the sort of squiggly lines that we'll talk about later, that is the chromatin, so that's kind of where your genetic information is stored, and then you have your nucleolus. So this nuclear membrane is a double membrane, meaning it's got two layers. The inner membrane is going to be supported by a protein matrix, and that's what's going to give the nucleus its shape. And then we've got these nuclear pores, these holes that actually run through both layers of the membrane, and they're allowing materials to move in and out of the nucleus. The nucleolus is, it says a dark staining area, so it's going to be like a darker area within the nucleus. There's going to be, sometimes none, sometimes up to 4 within a nucleus. And this is going to be a storage area for ribosomes. Then we've got chromatin. Chromatin is a mix of DNA and protein, and it will form the cell's chromosomes. So your cell's chromosomes are really only forming when your nucleus is dividing during mitosis. When that isn't happening, the DNA doesn't have to be organized in that way, and so it's in this sort of loose format that we call chromatin. And so the function of the nucleus is to essentially act as the control center of the cell. This is the area that contains all of your genetic instructions, because that's what holds the DNA. I mentioned ribosomes earlier, and so ribosomes, their structure is that they're made up of 2 subunits of protein and ribosomal RNA. There is no membrane separating the inside of the ribosome from the rest of the cell. And we're going to talk about ribosomes a lot in future chapters because they function in protein synthesis. So the information that's carried in DNA is then made into proteins with the help of ribosomes. Here you can see the structure of ribosomes. So again, they've got the 2 subunits, so they're very cleverly named, a large and a small subunit. So the large subunit is made up of about 45 proteins and then 3 ribosomal RNA molecules, whereas the small subunit is about 23 proteins and 1 ribosomal RNA molecule. Ribosomes are found all throughout the cell, and so they can be floating free in the cytoplasm, and those ribosomes are actually going to be making proteins that are used right there inside the cell. You can also have membrane-bound ribosomes as well, and they make proteins that are going to be exported from the cell. And so those are going to be proteins that are used in different areas. So the cell is actually going to produce those proteins, pack them up, and ship them out. And so they're going to be used somewhere else. So I've been talking a lot about membranes, and so we're going to focus now on something called the endomembrane system. And these are going to be membranes that are related through physical contact or continuity by the transfer of membrane segments called vesicles. And so if you look at this diagram, it's going to make a little bit more sense. So you can see we start with what we first talked about, which is the nucleus. Outside of the nucleus, you have the endoplasmic reticulum. Here we're looking at the rough endoplasmic reticulum. Over here is the smooth. And then on this side, we have the Golgi apparatus, also called Golgi bodies. And then we also have subvacuoles or vesicles floating within the cell as well. And so all of that is part of this endomembrane system. The first thing I mentioned after the nucleus was the endoplasmic reticulum. So a lot of times in class you're going to see this written as ER. And this actually makes up about half of the total membrane inside of the cells. And it's usually or often continuous with the nuclear membrane, meaning it's sort of connected to it. We actually have 2 types of endoplasmic reticulum that we're going to talk about, but essentially the structure, it's going to look like either folded sheets or sort of tubes of membranes. We say they're fluid in structure because they can change shape, and they do change shape pretty constantly. So again, we have the 2 different types, the smooth ER, which has no ribosomes, and this endoplasmic reticulum is going to be used for things like lipid synthesis, can help store carbohydrates. The rough endoplasmic reticulum, or rough ER, has ribosomes on the outside, so it looks rough, and that's why we call it rough endoplasmic reticulum. And this is going to be the part that actually makes those proteins that the cell is going to secrete or export so they can be used somewhere else. Next, we have the Golgi apparatus. You'll also see this written as Golgi bodies. The structure is going to be, here it says parallel array of flattened cisternae. Really, it looks like pita bread, sort of stacked together. Again, you're going to have 3 to 20 of these per cell. And they are likely an outgrowth of the endoplasmic reticulum system, and so they are usually very closely associated with that rough endoplasmic reticulum. And so here in the small diagram you can see the rough ER is right there, and then the highlighted portion is the Golgi apparatus. And so really it looks like stacks of pita bread. Alright, it's probably the most accurate description. There are 2 faces. There's the cis face, or the receiving face, And so that is going to be where the proteins are sort of received by the Golgi apparatus from that rough endoplasmic reticulum. And then there is the transphase, or the shipping side. And so this is where the side where we're getting rid of all those proteins that need to leave the cell. So the function of these Golgi bodies is processing, first of all. So it's going to take in the products from the endoplasmic reticulum, and it's going to modify them and get them ready to be sent out. And then the distribution is actually packaging them up for transport. So sort of like a fancy little post office within the cell, it's going to take in the products from the endoplasmic reticulum, modify them, getting them ready to send out, and then packaging them and sending them outside of the cell. There are also Golgi vesicles, and these are going to be like smaller little bits of the Golgi body. So these are going to be small sacs of membranes that actually bud off of the main Golgi body. And it's a transportation vehicle, so this would be like the mail truck that's then taking those endoplasmic reticulum products and getting them ready for transportation. Now, for whatever reason, the lysosome is my favorite organelle. And the lysosome is composed of a single membrane. It is made from the Golgi body, or the Golgi apparatus. And it's really important because it helps to break down or degrade cellular materials that are no longer needed. So these are little sacs that contain lots of different types of enzymes. We have over 40 types that are known, and they contain enzymes for fats, polysaccharides, proteins, nucleic acids. So they are like the cleanup crew of the cell. Go back for a second, and just like when we talked about the word hydrolysis and how we had that lyse part, hydrolysis was to break something apart using water. Here we have the lysosome, and we see that word lyse again, or that base lyse again, and so it really does mean to break down. Here we have just sort of a model for what happens with the lysosomes. So these are your lysosomes. You can see that they are budding off of the Golgi apparatus, and the little sort of dots inside are the enzymes which they contain. In this first example, you have this food molecule being brought inside the cell. That's called phagocytosis. That food vacuole is forming, so now the food is actually inside the cell. But just like we need to digest our food, cells need to digest food as well. And so in order to do that, you have a lysosome that's going to join with this food vacuole and break down the food molecule. Here is another example. This is autophagy, and so this is essentially, you can kind of tell it's like a little piece of mitochondria inside this vacuole. And so the lysosome, again, is going to connect to it. They're going to join together. It's going to release its enzymes, and those enzymes are going to break down that cellular structure that's ready to be degraded. Again, this is just another example showing you the exact same things with the food vacuole and the mitochondria and how they're both being digested. Here is your question. Tay-Sachs is a disease affecting infants. Those suffering have an accumulation of lipids in the brain that actually interferes with normal biological functions. So which of the organelles from this lecture do you think are involved with this disease? So I want you guys to send me an email to my CPS email account and give me your answer. Please make sure you explain why you say that particular organelle. So be specific. What organelles do you think are involved with this disease? And please explain why you think the organelles are explained in that disease. Alright guys, this was your first lecture, so you have to let me know what you think in class, and I hope you have a great day.